friends, welcome to my channel Star Biology Class. Friends, today we will discuss about rapid amplification of polymorphic DNA. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. First of all, let us know what is polymorphic DNA. The term polymorphic DNA refers to variations in DNA sequences that occurs naturally among individuals of a population. These differences are known as DNA polymorphism. These differences are known as polymorphism, which occurs because of different reasons. First one is single nucleotide polymorphism, SNPS. In this, one nucleotide pair of the genome is substituted by another different nucleotide pair. Number two, genetic variation also arises because of the presence of variable number of tandem repeats. Different individuals have different numbers of repetitive DNA sequences. As a result, DNA polymorphism arises. Number three, insertion and deletions or indels. Insertions or removal of nucleotides from the genome also creates genetic variation among individuals of a population. And finally, transposable elements or transposons, also known as jumping genes, move from one location of the genome to another location. Such movement of transposons also create DNA polymorphism. The term polymorphic is used when the sequence variation exists in more than 1% of the population. It is different from rare mutations. Now, what is rapid amplification of polymorphic DNA? Rapid is a quick PCR-based molecular biology technique that uses short arbitrary primers to amplify random sequences of DNA. In traditional PCR, two different primers, forward and reverse, are used to amplify specific DNA molecules. By contrast, in rapid, only a single type of short arbitrary primer having 10 nucleotides is used to amplify random sequences of different DNA samples. It is due to the presence of differences in the genomic DNA of different individuals. The single type of short primer binds to different complementary regions of different DNA samples. In next step, when the primer is extended by PCR using Taq polymerase, fragments of different sizes are generated. This figure illustrates the principles of rapid amplification of polymorphic DNA. In this example, we have genomic DNA samples from two individuals, sample A and sample B. As already said, in this technique, random sequences of genomic DNA are amplified using short arbitrary primer by polymerase chain reaction, PCR. PCR has three basic steps. First step is DNA melting, second step is primer annealing, and third step is amplification. All the three steps take place in the thermal cycle. Polymerase chain reaction requires a thermostable DNA polymerase such as tag polymerase, arbitrary primers, 4 different deoxyribonucleoside triphosphates and divalent magnesium ion. First of all, the genomic DNA sample is melted separately by increasing the temperature to 95 degrees centigrade. At such a high temperature, hydrogen bonds between complementary bases are broken and the DNA strands are separated. After melting, the complementary DNA strands, which are already separated, are annealed with the primer. Rapid amplification of polymorphic DNA, only a single type of short primer is used. Due to sequence differences between two DNA samples, because of all these reasons, the primer will bind at 3 complementary sites to both DNA strands of sample A. On the other hand, in case of sample B, the primer will bind at only 2 sites of both the DNA strands. These differences in primer binding is due to the presence of polymorphic DNA. When the primers are extended by tag polymerase, newly synthesized fragments of different sizes will be produced. The length of the newly synthesized DNA fragments will depend on the distance between two primer binding sites. This process of amplification is continued for several cycles. After amplification, the newly synthesized DNA strands are separated by agarose gel electrophoresis into distinct bands. You can observe that in case of sample A, we are getting 3 DNA bands, whereas in case of sample B, we are getting only 2. This is due to the fact that in case of sample B, at this region, the primer is not able to bind. This is due to the absence of complementary sequence in this region. This bending pattern of DNA of different samples serve as DNA fingerprint. These differences in DNA bending pattern of different individuals or this genetic fingerprint help us to assess genetic diversity, also in gene mapping and evolutionary studies, in population genetics, in diagnostics, for paternity testing, and also for plant and animal breeding. Rapid amplification of polymorphic DNA has several advantages. This technique is fast, efficient, inexpensive. It requires small amount of DNA. One of the main advantages is that it do not require any prior information of DNA sequence. Rapid has several disadvantages such as low reproducibility, and it is also not able to distinguish homozygotes from heterozygotes. Friends, this is all about rapid amplification of polymorphic DNA. For more updates, subscribe my YouTube channel, Sam Biology Class.